you studied the previous lectures? Who has studied the previous lectures? All of you? You, and you. Raise your hand if you have studied the previous lectures. Please raise your hand if you have studied the previous lectures. No? Just you.
Just two.
Just 2 lectures. It would be difficult if you do not study the lectures. And I told you, if you leave the lectures for the day before, or the days before the exam, or the night before the exam, you won't, or you cannot pass the exam.
Because I haven't enough time to study.
You've had the slides around 3 or 4 days before the lectures. And each day I have 3 lectures, and you had time from 3 p.m. until 12 a.m. It's around 9 hours. If you need to rest, 2 hours is enough for rest. And again, you will have 7 hours to study after the class. Yeah? So you need to study. You should put your cell phones away. You should put Instagram and other social media away. You have to study. You do not need to waste your time in social media. Which one? Doesn't matter. You're really wasting your time. Okay? Today, we will have 3 lectures. First of all, I'll talk about B cell development, and then T cell development, and indeed humoral immune response of adaptive immunity, and then T cell development and T cell activation and cellular immune response. And then I'll talk about tolerance mechanisms and a little about autoimmune diseases, basics of autoimmune diseases. Diagram is familiar with you again. Immune system, 2 layers, many layers of immune system: innate immunity, adaptive immunity, and you know that in innate immunity we have barriers beside cells. I have talked about the cells, and we will have a lecture about barriers or mucosal immune surfaces. And today I'm going to talk about adaptive immune system, both B cells and T cells, humoral immunity and cellular immunity. And generally, you know that humoral immunity is against extracellular antigens mainly, not only, and T cells mainly again, or cellular immunity is better to say, is against intracellular antigens, mainly, not only. You will see that we have some T cells that are involved in humoral immunity. And again, you will see that we have antibody against intracellular antigens. So we are talking about the most efficient part in each response. We cannot say that, for example, against viral antigens, that they are intracellular microorganisms, we have just T cell immune response. It's not so. We will have antibody production against viral antigens. So we have humoral immune response against viral antigen as well. So we have activation of both of them. But the response of T cell against intracellular immune response is more efficient, more effective, more protective, and responses of B cell against extracellular is more protective and effective. It's very important. I talk about B cells, B cell development, and the generation of BCR. How B cells will produce or can produce a variety of BCRs with different specificity. We are going to talk about this topic today. It's very important. B cell development, that we know it occurs in the bone marrow, and then I'll talk about B cell activations, and then effector mechanisms of humoral immunity. And you know that effector mechanisms of humoral immunity indeed are the functions of antibody. Because the final outcome of B cell activation is production of antibody. So anything that does antibody is indeed effector mechanisms of humoral immunity. For example, if I ask, let me ask the question at the end of the class. So, keep in mind, it's very important that effector mechanisms, you know this, effector mechanisms of humoral immunity is the functions of antibody. Activation of complement, for example, doing ADCC, you know them, acting as an opsonine, neutralization, and I'll talk about them again today in summary. I'll recall you them today in summary. So, first of all, I'll talk about B cell development, then B cell activation, and then B cell function, or indeed antibody function. You know that B cell development occurs in bone marrow. And then B cells, after activation, they leave the bone, after maturation, they leave the bone marrow, and in the lymphoid tissue, they wait for the antigen, secondary lymphoid organs. Indeed, bone marrow is a primary lymphoid organ, the site, the location for the generation of the B cells, and then secondary lymphoid organs, including spleen and lymph nodes and other secondary lymphoid organs, B cells wait there for the antigen. If there is antigen, then we will have a specific antigen, then we will have B cell activation and production of antibody. We are going to talk about, first of all, B cell development in bone marrow, because the B cell development occurs in bone marrow and generation of BCRs, B cell receptors, occurs in the bone marrow. When B cells are developing in the bone marrow, they will produce BCRs. And there is a same story for T cell. T cell development and then T cell activation. The development process, mainly production of TCR specificity or rearrangement of TCR genes, is very, very similar to BCRs. So we have same process for T cell development and B cell development. But you know that this T cell development occurs in the thymus, another primary lymphoid organ. And again, mature T cells leave the thymus and go to the secondary lymphoid organs, lymph nodes and spleen and other lymphoid organs, and wait for the antigen. If there is antigen, we will have T cell activation and their function. First of all, I will talk about B cell development. Do you have any question about these general topics? B cell development, humoral immunity, extracellular antigens, intracellular antigens. Do we have antibody against intracellular antigens? Is that possible to have antibody against intracellular antigens? Yes? What do you think? She says yes. What about you? Don't you think about it?
No, because the antibody cannot
She says no. We do not have antibody against intracellular antigen, for example, a virus, because antibody doesn't have access to the inside of the cells. What else? What else? for viral antigens, viral infections, or intracellular pathogens, for example, the final destination of them, or the final living site or location for them is intracellular spaces. I'm talking about intracellular pathogens, microorganisms. The final destination, for example, an HPV lives inside the hepatic cell. Is that correct? HPV, hepatitis B virus, lives inside the hepatic cells. So its final destination, final location is inside the hepatic cell. But after infecting this hepatic cell, the virus will kill the target cell and it will release to the interstitial fluid and to the blood. And then it will infect new cells. So we have this virus in this space as well. So viruses or intracellular microorganisms, they are in the extracellular spaces transiently in order to infect new cells. Okay? So when they are in extracellular spaces, we will have antibody against them. So we have antibody production against intracellular pathogens because in their lifespan, in their life cycle, they have a time that they are out of cells. They are exiting from one cell and go to the another cells. So they are transiently exist in extracellular spaces. Is that clear or not? The question was this: do we have humoral immune response or antibody production? They are same topics against intracellular pathogens or not? Again, do we have antibody against intracellular pathogens or humoral immune response or not?
No, we have.
Yes?
Yes.
Yes? Yes. I told you. For example, I gave you the examples of HPV. HPV is a virus that infects a hepatic cell and lives inside the hepatic cells. But when we have a proliferation of the virus inside this cell, it will kill the target cell and it will release to the outside of the cell, to the interstitial fluid, to the blood. So they are transient in the blood to infect a new cell. So sometimes we have some viruses outside of the cells because they are passing from one cell to another cell after killing of the first cell. Okay, clear or not? So when they are in the extracellular spaces, we will have antibody production against them. Now antibodies have access to those viruses. So we have antibody production against viral antigens. That's why we say one function of antibody is neutralization of viral infection, neutralization of viruses. Neutralization, I told you, means binding of antibody to these viruses and prevent the binding of those viruses to the new cell, to protect the healthy cell, the normal cell. Yeah? Got it? So again, do we have antibody production or humoral immune responses against intracellular pathogen or not?
Yes.
Do we have T-cell response? Of course, yes. Of course, yes, because they are intracellular pathogens. So we have both T-cell response and B-cell response. Both of them. We have both of them. But T-cell response is more protective, is more effective. That's why we say the T-cell response against cellular immune response against intracellular pathogens is more effective. Is the main cells that can protect us from that virus. mean that we do not have B cell immune response. Of course we have. Clear? No question? So, B cell development, I told you, occurs in bone marrow. And in the bone marrow, from a stem cell, hematopoietic stem cell, these cells develop in sequential stages from one stem cell toward a mature lymphocyte. This image is similar for both T cells and B cells, but the difference is that T cells development occur in the thymus, but B cell development occurs in the bone marrow. The story is same, the process is same. We have a stem cell and some intermediate immature cells and then a mature cells. So they are developing, they are maturing step by step over time. And in this development process that occurs step by step over time, we have some important events that mainly produce the receptors, BCR for B cell, TCR for T cell. And that's why today I'm going to talk about initiation and antigen receptor gene rearrangement. It's very important a story or topic in immunology. How B cell can produce millions of TCR with different specificities. I'll talk about it. And then in the third lecture, I'll talk about selection of the repertoire or indeed tolerance mechanisms. I'll talk about both of them. And you can see here that before maturation, the developing T cells and B cells are not exposed to non-self antigens. They are only exposed to self antigen. I have told you about this, and I'll talk about it more in the third lecture. So before maturation, that B cells or T cells are not mature, they are immature cells. They are not exposed to non-self antigen. In the bone marrow and in the thymus, we do not have exposure to foreign antigens, non-self antigens. We are the T cells and B cells are only exposed to self antigens to check if they are self-reactive or not. I'll talk about it completely in the third lecture. Clear or not? But after maturation in the secondary lymphoid organs from here, secondary lymphoid organ, they are exposed to both self and non-self or foreign antigens in the lymph node spleen. So during development, in the primary lymphoid organs, bone marrow or thymus, we do not have any exposure. It's very important to non-self or foreign antigens. They are only exposed to self antigens. And after maturation, they go to the secondary lymphoid organ and wait for the specific antigen. It could be self or non-self antigen. Is that clear? Yeah? So the process is same for both T cells and B cells. As I recall, this is an antibody that here those chains are bound to each other and here light chain to heavy chain. So this is the structure of antibody, two same heavy chains and two same light chains. And this part is no. This part. Antigen binding site, which is composed of two variable domains of light chain and heavy chain. Okay? It is the antigen binds here, for example. And antibody has two antigen binding sites. This one is this part, this R. Each R is called FAB or FAB, fragments of antigen binding. And this part is called FC. And you know the functions of antibody, that most functions of antibody are related to FC, including activation of ADCC, activation of complement, as an upstream, all of them are related to FC. Or transferring of antibody between mother and fetus, transferring of antibody from blood to the mucosal surfaces, all of them are done by FC, that with binding of them to FC receptors. And this is a B cell, and they are again antibody. What's the difference between these two antibodies? What is the difference between these two antibodies? Free antibody and cell-bound antibody.
Cell-bound antibody is bound to the surface of the B cell, but the antibody is in the serum.
Yes. The only difference between those two antibodies is that this one is free in the serum, in the plasma, in the body fluids, but this one is bound to B cell. That's it. Indeed, they have two variable domains here and constant domains and FSC, but they are bound to B cell. When they are bound to B cell, we call it BCR. Yeah? So BCR is indeed, we can say that the BCR is a B cell bound antibody. That's it. Is that clear or not? Please, if you do not understand, tell me, explain again. So, on the surface of B cell, there are so many BCR, not just one. You can see it here. On the surface of B cell or T cell, it doesn't matter. It's the same story again. We have several or many, not several, many B cell receptor for B cell or T cell receptor for TCR, not just one. Okay? It's very important to know that I told you that one antibody has two antigen binding sites. Does the specificity of this antigen binding site different? Is the specificity of this antigen binding different or not? It's same. Both of them are against same epitope. For example, this one if is against A, this one is against A. Because the composition of them is same heavy chain and same light chain. So one antibody is said that is monospecific, but divalent. It can bind to two epitope with its two arms, but it is monospecific. And all the BCRs of one B cell, I'm talking about just one B cell. All the BCRs of one B cell, all of them are against just one epitope. I'm talking about one B cell, not the whole population of B cells. So I told you that the B cell has many BCRs on its surface, and all of these antibodies are against just one epitope. Yeah? Clear? For example, this one is against A, this one A, this one A epitope. It's the name of epitope. All of them, it means that this antibody is not against A, and this one, for example, epitope B or epitope C. No. All the antibodies, all the BCRs on the surface of one B cell, all of them are against just one epitope. So, two antigen binding sites of one antibody against one epitope, the same epitope, and all BCRs on the surface of B cell, again, all of them are against just one epitope. Here, epitope A. That's why we say that, for example, this B cell is anti-A B cell. That T cell, the story for T cell is same. That T cell is anti-X, for example, anti-Y, anti-Z T cells. Is that clear? It's very important. No question? So, until now, in summary, I told you, two antigen binding sites of one antibody against one epitope, and all BCRs of one B cell against one epitope. But inside the body, we do not have just one cell against one epitope. Keep in mind, we do not have just one cell. For example, inside my body, I do not have just one B cell against COVID antigen. I have many B cells or several B cells against each epitope. So when we have a population of cells, all of them are B cells, that they are distributed in the body, but all of them have same PCR. It doesn't mean that all B cells have same BCR. A population, a small population of B cells, all of them have same BCR. It's logical. It's not logical to have just one cell against each epitope. It cannot induce a strong immune response. We have several B cells or many B cells against each epitope. Okay? So this population of lymphocytes, both or T cell or B cell, it doesn't matter. This population of the same lymphocytes, when we say all of them are against or have same BCR, it means all of them are against same epitope. Thank you, yes. All of them are against just one epitope. So the BCRs of all of them, we know that on the surface of them there are so many BCRs, and BCRs of all of them are same. All of them have same BCRs, several BCRs on them, and all of them against one epitope, one specific. That's why we say immune response or B cells are specific. So this population is called a clone of lymphocytes. A clone of lymphocytes. So clone means a population of B cells or T cells. That all of them, a population, all of them are against just one epitope. Indeed, all of them have same BCR or TCR for T cell. Clear? No question? So, again, one antibody, two antigen binding sites, both of them against one epitope. One B cell, many BCRs, all of them against, and many of these B cells, all of them same BCR against one epitope. And this population is called a clone of lymphocyte. And again, all of the members of this clone, indeed clone is a group of B cells. That all of them are against just one epitope. So all members of this group is against just one epitope. No question? Inside the body, we have 10 to the power of 7 and sometimes 10 to the power of 9 different clones of B cell in the body. Millions of clones there are in the body. What does that mean? There are millions of B cell clones and T cell clones in the body. Sometimes a billion. Some textbook says that few billions. What does that mean? Think about it. Diversity. Yes, diversity against what? Yes, it means that when we have 10 millions of clones, we are talking about different clones, different groups. One group, second group, third group, and millions group. We have millions of group of lymphocytes that This group is called clone. So we have millions of clones. Indeed, each clone is against just one epitope. So we have millions of specificity for T cells and B cells. Millions of specificity. And sometimes few billions. Think about it, it's very important. All of us have this huge diversity of clones in our body that they are waiting for antigen. That's why, for example, before COVID-19, we haven't been exposed to COVID antigen in our life. But after entrance of COVID, we can produce immune response because we have millions of clones that some of them are against COVID antigens, or some others are against HPV antigens, some others against E. coli, some others against schistosoma, some others against any antigen. Okay? It means a specificity or diversity. That's why we say immune response, immune cells, or we have a huge specificity in adaptive T cells or B cells. So again, let's begin the story from the beginning. One antibody, two antigen binding sites, one epitope. One B cell, several BCRs, all of them against one epitope. And we have a population of this cell, a group of this cell, all of them same BCR, and all of them against same epitope. This is called a clonal lymphocyte. And again, we have millions or few billions of T cells or B cells against millions or billions of antigens. Okay? Now, we are going to talk about how immune system can produce millions of BCRs or millions of TCRs. The process that enables T cells or B cells that are developing in the bone marrow and, or the thymus respectively to produce millions of TCR or BCR. Today I'm going to talk about this. No questions? Again, another figure for antibody, constant regions, variable regions, two variable regions, antigen binding site. And for example, they are different epitopes. We have different variable regions against each, and indeed the shape of each variable domain, the shape of each variable domain of different clones is in a way that, for example, can only bind to this epitope, not other epitope. Okay? And the shape of, for example, this one is in a way that cannot bind to this one, or to this one, or to this one. That's specificity means this. It's I told you, it's similar to lock and key. This one is lock, this one is the key, this one is lock. Doesn't matter which one. So I need to, before going to the process of production of millions of different BCR. It's important that I'm telling you that we have millions of BCR or millions of TCRs inside the body, or millions of species. When we say millions of BCR, the only part of the BCRs that is different from each one is variable regions. That's why we say this part variable region. We have a variety of those regions that each one is specific to a specific epitope. Okay? So when we say we have millions of BCR or millions of TCR, the difference between them is just in variable regions. Those parts. Only we are going to talk about those parts. Variable regions. The variable regions determine the specificity of the antibody. The constant regions have nothing to do with specificity or binding to different epitopes. We are just going to talk about, when we talk about specificity, when we are talking about the fitting of the shape of epitope with antibody, we are just talking about variable region, antigen binding site. That's logical because this part binds to epitope or antigen, and those parts have other functions. They do not bind to the antigen. So, before talking about the process of production of millions of BCRs or millions of specificity in T cells and B cells, I should remind you some basics of biology. You know that inside a cell we have a nucleus, and inside nucleus we have chromosomes. 23 pairs of chromosomes are in the nucleus of each cell, and you know that chromosomes is a dense, a packet of DNA. The DNA is packed in the chromosome, and chromosomes are in the nucleus of the cells. And the sequence of on the sequence of DNA, we have different genes. That each gene will produce a specific protein. Okay? You know this. Cell, nucleus, genome. The whole content of the chromosomes of cells is called genome. So we have the DNA that is packed on the chromosome, and on the DNA we have different genes that each gene will produce one protein. For example, albumin, there is a gene for albumin, there is a gene for the complement protein, there is a gene for example, interleukin 2, there is a gene for TNF alpha, and so many proteins are produced in the body, and for all of them there is at least one gene. Okay? Clear? There are chromosomes. Thousands of genes are distributed in 23 pairs of the chromosomes. So, we have 23 pairs of chromosomes, and just there are around 20,000. Please, there are just 20,000 different genes on the chromosome. 20, sometimes it is said 30,000. So around 20 to 30,000 of genes we have on our chromosomes. Just 20,000 or 30,000. So it means that capacity of the genes to produce millions of TCRs is very low. There is two concepts here. The first one is that we have just 20 to 30,000 genes, different genes, but we need millions of TCR from few proteins, few genes. There is a process that enables T cells and B cells produce millions of different TCRs and BCRs from few or thousands of genes, not few, thousands of genes. There is a process that enables T cells to produce millions of TCR. I'm going to talk about this process. Yes, by DNA rearrangement or DNA recombination, that I'll talk about it. Increase the, yes, the variable specificity. No, I'll tell you. Is that clear? So we need at least 10 million antibodies or BCR or TCR. Indeed, we need 10 million clone or 10 million BCR. You know this. 10 million BCR means 10 million clone. So at least we need 10 million clones or BCRs. But just we have 20 to 30 thousands of genes. So we need some process. So one gene for one receptor cannot produce millions of TCR or BCR. we need process to produce such a huge specificity of TCR and BCR. No question, no comment. So you can just please 10 minutes. 10 دقایق, not more.